Children, Saira, welcome to this session. Today we are going to continue with lesson number 2, Life Processes in Living Organism Part 1, Revision Part. Okay, already we have started with the lesson and I have finished half of the lesson. Now let us continue with the next part, which is the cell division. Already we have learned there are two types of cell division. One is mitosis and the second one is meiosis. Okay, mitosis occurs in somatic and stem cells and meiosis occur in germ cells. This explanation I have given you in the previous video itself. Okay, and mitosis is completed by two main steps. What are the two main steps? Karyokinesis and cytokinesis. Karyokinesis means nuclear division and cytokinesis means cytoplasmic division. Okay, karyokinesis is completed in what are the four steps? Prophase, metaphase, anaphase and telophase. Okay, by now you should be thorough with this lesson, but let me revise this again. So what are the four steps? Prophase, metaphase, anaphase and telophase. So in prophase, what happens to the chromosomes? They become condensation of basically thin thread-like chromosome starts. Usually the chromosomes, which is before this prophase, interphase is there, then the chromosomes number will be doubled up, and when it is thick and long, it will become, in this prophase, the condensation will start. Due to this, they become short and thick and they start to appear with the pairs of sister chromatids. Sister chromatid means the same type of chromosomes. Okay, they will appear in the pair with the sister chromatids. Then centrioles duplicate and each centriole moves to the opposite poles of the cell. Okay, I have shown in the video which I have sent in the group. Just go through that. You can see the process very clearly. Nuclear membrane and nucleolus start to disappear. Okay, you can see this prophase. You can see the chromosomes, how they appear with the sister chromatids. And the centriole is duplicated and it is going to the opposite poles, and the nuclear membrane, you can see the nuclear membrane also slowly starts disappearing. Okay, the next comes the metaphase. In metaphase, nuclear membrane completely disappears. Okay, and the chromosomes complete their condensation and become clearly visible along with their sister chromatids. So the nuclear membrane disappears and they are clearly visible with their sister chromatids in metaphase. And main point of this metaphase is the chromosomes are arranged parallel to equatorial plane of the cell. You can see in that video which I had sent how in metaphase they come and stand in the middle of the cell, that is in the equatorial plane, and special type of flexible protein fiber called the spindle fibers are formed between the centromere and each chromosomes and both the centrioles. You can see that in that video which I had sent. Okay, that is the spindle fibers are formed, and that is, you can see the spindle fibers like this and how they are arranged in the equatorial plane in the metaphase. Okay, in anaphase, centromeres split and thereby sister chromatids of each chromosome separate and they are pulled apart in opposite directions with the help of spindle fibers. This, there is the pulling of spindle fibers occurs, and the sister chromatids are divided into two parts. Okay, so one half goes up and the second half comes down. Okay, separated sister chromatids are called as daughter chromosomes. Chromosomes being pulled appear like bunch of bananas. You can see this is the characteristics of anaphase. You can see where it looks like the chromosomes appear like bunch of bananas. You have to write anaphase. In this way, each set of chromosomes reach at two opposite poles of the cell. Okay, so you can see the diagram how their spindle fibers are pulling both the sides and they are divided into two parts. The chromosomes are divided, one half goes up and one half comes down. Okay, in telophase, that chromosomes which have reached the opposite poles of the cells now start to decondense, due to which they are again become thread-like and thin and invisible, and nuclear membrane is formed around each set of chromosomes reached at the poles. After this, the nuclear membrane starts to appear and it becomes thin and long after this, because it was condensed so much. So in all these phases, when it comes to telophase, slowly it starts to again it becomes thin and long, and thus two daughter nuclei are formed in the cell. and nucleolus also appears in each daughter nucleus, and spindle fibers completely disappear. In this way, karyokinesis completes and cytokinesis starts. I told you cytokinesis means the cytoplasm divides into two. Okay. So after this karyokinesis, which has got 4 phases, 4 steps, prophase, metaphase, anaphase and telophase, this cytokinesis begins. Inside, okay, this is the telophase. You can see there are two, it become thin, condensation is over and now it become thin and long. Nuclear membrane has also started appearing. Everything is there, but it is connected to each other. Then construction in the cell membrane, there is cytokinesis taking place. Constriction takes place in between the cell, and you can see here two daughter cells are formed, which is of similar type. There is no change in each cell because one, it is divided into two cells, so cytokinesis takes place and two daughter cells are formed. Okay, it is why this mitosis is essential. It is for the growth of the body, it is for the restoration of emaciated body, wound healing and formation of red blood cells. This mitosis is very essential. I think mitosis is very clear. Now let us go to meiosis. Meiosis is not explained more in your textbook. What is there I have explained, and some of them I have taken from the other books, and I have sent the videos. You can see the videos for the meiosis 1 and 2. Okay. Now it is completed in 2 stages, meiosis 1 and meiosis 2. Okay, mitosis is only 1, but meiosis, there are 2 stages, meiosis 1 and meiosis 2. Okay, under meiosis 1, there is recombination or crossing over between homologous chromosomes. Homologous chromosomes means the chromosomes from each pair, one is from the male and one is from the female. And thereafter, those homologous chromosomes are divided into 2 groups, and thus 2 haploid cells are formed. Okay. So at the end of the meiosis 1, how many haploid cells are formed? 2 haploid cells are formed. In that video which I had sent, in that meiosis 1, all the process is explained very well, the 5 phases of all those. So this meiosis 1 has prophase 1, there are 5 stages, prophase 1, metaphase 1, anaphase 1, telophase 1. Okay, and as usual, you can see here, under prophase 1, there are 5 steps. And they are, what is that? It is very lengthiest process of prophase 1. There are 5 parts of it. Under that first step is Diplotene, zygotene, Pachytene, and you can see all those 4, 5. Okay, well, let us start with the Diplotene. Diplotene, in this condensation, makes chromosomes become distinct and compact. You can see here, this part is not explained in our textbook, but as a question is asked behind, I have given this from other books. Okay, you can write it down and you can learn this only. Condensation makes chromosomes more distinct and compact. Then nucleus enlarges, and nucleolus and nuclear membrane starts disintegrating. Okay, it becomes thick and short. And the next one, zygotene, homologous chromosomes starts pairing together by the process called synapsis. Okay, that is, I told you homologous chromosomes means one from the male and one from the female, and they pair together by the process called synapsis. That is very well shown in that video. 2 synapsed homologous chromosomes form a complex called bivalent or tetrad. Okay, it is called as bivalent. When they are synapsed together, they are called as bivalent. Okay, then the Pachytene is the 3rd stage. It is the longest phase of prophase 1. Then recombination nodules appear in the stage at the sites where crossing over has to be taken place between non-sister chromatids of homologous chromosomes. That means the nodules are formed when they are paired together. There is at that place nodules appear in the stage, and at the sites where crossing over is going to take place in that place. Okay, between the non-sister chromatids, between 2 pairs, different. So that means of the homologous chromosomes. Then the Diplotene, the synapses break down and the chromatid starts repelling from each other and begin to separate. Okay, in Diplotene. And in diakinesis, the chromosomes are fully condensed the nucleolus and nuclear membrane dissolve, and the mitotic spindle begins to assemble. The spindle will start appearing in the metaphase, isn't it? Even in the mitosis, in the metaphase, the spindle fiber will start appearing. So this prophase of meiosis 1 has got 5 stages in this. All these 5 stages, there is crossing over of homologous taking place, and then when you come to metaphase 1, slowly the spindle will start coming up. Okay, you can see this Diplotene, zygotene, Pachytene. You can see how they are nodules are formed, tetrad. Then Diplotene crossing over has already taken place, and Karyokinesis you can see like this. Then after this, there comes the metaphase. You can see the metaphase. The same way spindle fibers are formed, and they are arranged in the equatorial plane as it is in the mitosis. Okay, these are the 5 phases which is given in your textbook. You can see this figure, and also I have shown you some other figure also. Then in anaphase also, you can see here the same way it is pulled apart both the sides, but in chromosome the chromatids are separated. Here it is, you can see there is a difference in the color combination also. You can see anaphase 1, and in the end of telophase 1, you can see 2 sets have come this side, but all are of different types. They are not of same type. So 2 daughter cells are formed at the end of telophase 1. This 2 daughter cells will take part in the meiosis 2. Okay. In meiosis 2, the 2 daughter cells which is formed in meiosis 1 will take part in the cell division. Okay. In this stage, the 2 haploid daughter cells formed in meiosis 1 undergo division by separation of recombined sister chromatids. Already the sister chromatids are together. They are going to separate now. And 4 haploid daughter cells are formed. So, they are already in pairs. Then now again there is separation of the sister chromatids. And after that, you can see that there are 4 pairs, 2 pairs here, and that will divide, and you will get 4 haploid daughter cells at the end. You can see here how the prophase 1, as you can see prophase 2, this is of meiosis 2. Prophase 2, you can see that there are 2 parts. Then again metaphase 2, in both the cells you can see there are spindle fibers formed, and they are arranged in equatorial plane. And you can see in anaphase 2, they are pulled apart. If only one set has come, see half of gone up and half of the chromatids has come down, and at the end you can get in telophase there are 4 sets of haploid daughter cells. Okay, so in the video it is nicely explained, and you can visually see what is the how the meiosis takes place. Meiosis 1 and meiosis 2, what are homologous chromosomes, color difference is shown there. You can see how they becomes tetrad, and what is synapsis, everything it is shown there. So visually when you see that video, you will understand much better. So after seeing this, you see that video, so it will be even more clear. Hope this cell division is clear, children. So we learned today about mitosis and the 4 karyokinesis. There are 4 stages of karyokinesis: prophase, metaphase, anaphase and telophase. And after telophase, then comes the cytokinesis, that is cytoplasmic plasm divides into 2 parts. Okay, then you get 2 daughter cells at the end. But in meiosis 1, prophase 1 has got 5 steps: Diplotene, zygotene, Pachytene, Diplotene, and then diakinesis. 5 steps are there. You have to learn all those. Only those 5 steps. Then metaphase 2. Then you get metaphase 1, prophase, anaphase 1, telophase 1. In meiosis 2, there are 5 steps. We saw that. Prophase 2, metaphase 2, anaphase 2, and telophase 2. So this detail is not given in your textbook. So how much is there in your textbook, that much is enough. Okay. Hope you all understood, children. See you in the next video. Thank you, children. Bye bye.